Hi, this is Eric Lyons, and today's tutorial is going to use CoGe in order to identify and visualize differences between assemblies of the maize genome. This tutorial is applicable to any organism in CoGe for which there are multiple assemblies. We're going to begin the analysis using SynMap, which is CoGe's tool for generating whole genome syntenic dot plots. SynMap requires you to identify two genomes, and we're going to search for a maize genome by typing in maize in the name search box. This will return any organism whose name contains the word maize. At the bottom of this list is the maize. When this is selected, we'll now have a list of all the genomes for that particular organism. Maize has many genomes that are available. We're going to first select version 2 of the maize genome assembly using filtered genes, and the primary sequence has been masked. For organism 2, we're also going to search for maize, and we're going to select it from the bottom of the organisms that match, and we're going to now select version 1 of the maize genome assembly with filtered gene sets that have also been repeat masked. Once these genomes have been selected, select or press Generate SynMap in order to do the whole genome comparison. When the results are returned, you will have a syntenic dot plot where every syntenic gene that has been identified is colored green. As we look across this dot plot, we'll see that there is a strong line running up the 45-degree axis. Each one of these black horizontal or vertical lines separates one chromosome from another, and we can see that we have a very strong syntenic signal for every pair of chromosomes. However, you'll notice that there are many other syntenic regions that have been identified. This is due to identifying homologous or paralogous syntenic regions from the maize-specific whole genome duplication event, as well as very small syntenic regions from the grass-specific whole genome duplication event that happened prior to the radiation of the grasses. You'll also notice that as we look across this syntenic dot plot, that the chromosomes are ordered not by their name, but by the physical size of the chromosome. What we're going to do is we're going to modify the analysis in order to first sort all of the chromosomes by name, and then we're going to put down a filter that will only save the self-self syntenic matches and remove all the other matches, syntenic regions from this analysis. So under display options, we're going to sort the chromosomes by name. Under the analysis options, we're going to turn on an algorithm called quota align for enforcing a specific ratio of coverage between these two genomes. In this case, we're interested in identifying the syntenic regions that give us the best coverage once between these two genomes. So we need a syntenic depth coverage of 1:1 between these two genomes. When the results are returned, we'll see now that we just have that nice 45-degree line, all the chromosomes are ordered by size, and we've removed all of the other syntenic regions from this analysis. Next, we're going to add a visualization filter that's going to color inversions differently than that for regions that are not inverted. This will result in regions that are inverted being colored blue. When the results are returned, we can see inversions where we have blue lines as well as little tiny blue dots within these. If we want to visualize one of these chromosome by chromosome comparisons in more detail, we can click on that particular box and we will get a zoom in of just that comparison. As we look at this, we can see that the overall structure of the maize genome is highly similar between these two assemblies. There are few regions where we've had a large-scale inversion and a couple spots where pieces have moved between the two assemblies. When we zoom in, we can see in much greater detail the kinds of changes that have happened. So this is one of the cases where we have regions that have moved around relative to one another, just between the two assemblies. Now, this relatively high degree of concordance between maize versions is due to the back-by-back sequencing approach that was used for the maize genome. However, within each one of these backs that was shotgun sequenced, there are many changes that have occurred. In order to visualize these, you can use the crosshairs and move over any region that's synthetic. When the crosshairs turn red, this means you're on top of a synthetic gene pair, and when you click on it, it will launch CoGe's tool GEVO that permits high-resolution analysis of two genomic regions. When linked from SynMap, the gene pair on which you clicked is used to anchor genomic regions based on where that gene lies. You then have the ability to select additional sequence upstream and downstream from that gene for comparison. For this example, we're going to do a couple of modifications to the default analysis. The first is that since we're comparing the maize genome to itself, and the maize genome contains many repeat sequences, primarily from rampant transposon activity, we're going to mask all regions of the genome that don't code for protein. To do that, you want to open up the Sequence Options menu, either by clicking on Sequence Options or on the Open All Sequence Options, and under Mask Sequence, select Non-Coding Sequence. Next, we're going to compare a larger amount of the region than the default 50,000 nucleotides. In this case, we're going to compare 2 megabases upstream and downstream from each one of these genes, for a total of 4 megabases of the genome sequence. GEVO dynamically will extract these regions, mask them, and then do the comparison for you using the default algorithm of BLAST-C. When the results are returned, each one of these panels represents one of the genomic regions. The background that's colored purple represents mask sequence, and those that are green represents coding sequences as these little tiny arrows. All regions of sequence similarity that have been identified get a pink block. When you click on these, it will draw a line connecting regions of sequence similarity to one another. To highlight all of them, hold the Shift key and click on one of those pink blocks. If the blocks are drawn above the gene models, this is when the regions of sequence similarity are in the same orientation. When they're drawn below the gene models, this is when they're in the opposite orientation and one of these regions has been inverted with respect to one another between the assemblies of the maize genome. From this we can see that overall there is a high degree of concordance in terms of the gene order, but there are a couple of regions that have been inverted with respect to one another with the new version of the maize genome. That concludes this tutorial on how to visualize differences between assemblies of the maize genome. Thank you.